Next application of biotechnology is production of genetically engineered insulin. Insulin is used for diabetic patients and it was uh, extracted earlier from the pancreas of slaughtered pig and cattle. Because these are the organisms which belong to different species, इनको लेने से the patients they suffered from various allergic reactions and they developed reactions to the foreign proteins. Insulin is a protein which consists of two short polypeptide chains, chain A and chain B, and these chains are linked by disulfide bridges. This is the structure of insulin molecule, where we can see this is the chain A, blue one, and this green one is the chain B. These two are insulin chains which are joined by S and S, that is the disulfide bridges, and we can see the short chain A and B, they are joined together by an extra segment of protein which is called free peptide. इंसुलिन जब प्रोड्यूस होती है बॉडी में, वो किस फॉर्म में प्रोड्यूस होती है? एज प्रो इंसुलिन, इनएक्टिव फॉर्म में, यानी कि टू चेन्स chain A and chain B, they are joined by disulfide bridges, and in between them there is present an extra peptide chain which is free C peptide. When this insulin matures, means it becomes active or comes in functional form, तो ये जो free C peptide है ये उससे अपने आप separate हो जाता है and it becomes the functional insulin. Now what is done in genetically engineered insulin? The gene for coding this polypeptide chain A and gene for coding this polypeptide chain B, they have been isolated. These genes, they are isolated, they are introduced into the separate bacteria. उनमें क्या प्रोड्यूस हुआ? चेन ए अलग प्रोड्यूस हुई, चेन बी, पॉलीपेप्टाइड चेन बी इज प्रोड्यूस सेपरेटली. These two chains are produced separately and then they are joined by the disulfide bridges and then they are used as insulin. Let us see, in human beings, insulin is synthesized as prohormone. It needs to be processed before it becomes mature and functional. It contains an extra stretch called C peptide, and C peptide is not present in mature insulin and it is removed during maturation. Now, the main challenge for production of insulin using recombinant DNA technology was getting insulin assembled into mature form. इसको solve करने के लिए क्या किया? In 1983, Eli Lilly, an American company, it prepared two sequences of DNA corresponding to chain A and B. Chains of human insulin were then introduced into plasmids of Escherichia coli. These chains were produced separately, then they were extracted and combined by creating disulfide bonds and to form human insulin. This is how the genetically engineered insulin is prepared.